Hello, microbiologists, and welcome to Chapter 20. Chapter 20 focuses on the gram-negative bacilli of medical importance and also begins the content for our third exam in the course. I do want to just preface my conversation with you today and let you know that this is going to be a very tough stretch of the course. And it's not so much that the information becomes difficult, but it's that there is a lot of information to keep track of. Over the next 4 to 5 weeks, we are going to be unpacking different groupings of microbes. So it's going to be really important for you to organize your notes. I even suggest creating a table and organizing some of the information for each of the different microbes that we talk about in the chapters. When I started teaching this course back at Goodwin 14 or 15 years ago, we actually organized the microbes in a different way. We used to talk about them by body system. And that was often confusing because we would talk about 4 or 5 different classifications of microbes within one chapter. When we switched to the current Polaro text, that was actually much nicer in terms of unit and material organization, in the fact that now all of the microbe topics are organized by the morphology and gram reaction of those bacteria. So today, we are going to start by talking about the gram-negative bacilli. And remember, bacilli are rod-shaped microorganisms. So everything that we talk about today falls into this gram-negative bacillus category. So we are going to focus today primarily on the aerobes. So these are the oxygen-using, respiring gram-negative bacilli. And we're going to focus starting-wise on the non-enteric bacilli. So this is a extremely large group of bacilli that do not form endospores, and they're found in a wide range of environmental habitats. Everything from the intestinal tract of animals to things like the respiratory tract, soil, and even in some cases, food and water. Most are really not medically important, but there are some true pathogens, and others are opportunistic, meaning that they take advantage of the body when the immune system has been weakened. And these have, since we're talking about gram negatives, we know that they have that extra layer in their cell envelope known as the outer membrane. And the lipopolysaccharide or LPS component of that outer membrane serves as a toxin, as an endotoxin. So we're going to get into a few different groups today. There's 6 total that we are going to look at. We have the Pseudomonas and Burkholderia, which are both opportunistic pathogens, meaning that they take advantage of the body when it's weakened. We have Brucella and Francisella, which are both zoonotic pathogens, meaning that their reservoir is from animals. And we also have Bordetella and Legionella, which are both mainly human pathogens. Bordetella causes pertussis, and that's also known as whooping cough. And Legionella pneumophila causes a respiratory disease, and we'll get into that in a little bit, but it's known as Legionnaire's disease. So, Table 20.1 in your book breaks down the organization of this chapter. Again, everything that we're going to talk about falls into the gram-negative rod category. So they are going to stain pink when we perform that differential gram stain on the cells. And then we can break these into 3 additional subcategories. We have one that fits into the obligate anaerobes, meaning that they are not able to survive in the presence of oxygen. The majority that we'll see fit into the aerobes, and we also have some that are facultative anaerobes, which means that they require the oxygen but can survive if oxygen is absent. We then, from there, take our facultative anaerobes, and we can talk about those that ferment lactose and those that do not ferment lactose. So our lactose fermenters include things like E. coli, Klebsiella, Citrobacter, and Enterobacter. So we're going to get into and talk about the majority of these groups today, but this is just a overall 30,000 foot view of the different microbial pathogens that we will get into as we talk about our gram negatives today. So again, table 20.1, this is just the text way of organizing this. I'm not going to go through these slides. Again, it's just to give you kind of an overview of some of the things that we're going to talk about today. I do like the systems profile for table 21, which walks through the different organisms and then also the particular targets to the body systems that they impact. So we are going to go through all of this information today as we break down each microbe. So we're going to start first talking about the pseudomonads. Specifically, we're going to talk about Pseudomonas, and this is actually a really interesting organism in that it has a lot of different characteristics to it. These are extremely small gram-negative rods. They are motile. They do possess a single polar flagella for movement. And the majority are free living. We find them in places like soil and water. And they're also able to colonize plants and animals. They are very important in the decomposition process, and they can also be used to alleviate environmental hazards through the process of bioremediation. We do find them as a nosocomial infection, meaning that they are often acquired in the clinical setting, and they produce a series of enzymes, oxidase and catalase. Remember that catalase was an enzyme that we saw allows microbes to break down toxic oxygen radicals into things like atmospheric oxygen. And we are going to see that they produce these water-soluble pigments. And these pigments are important because they provide resistance to antibiotics, and they also are pathogenic and kill off other microbes that grow near the Pseudomonas. So Pseudomonas aeruginosa is the major species that we're going to talk about. It's a common inhabitant of the soil and water, and it's extremely resistant to things like antibiotics, soaps, detergents, and dyes. It is, as we mentioned, it's a nosocomial infection in the fact that it is able to create biofilms, those very thin layers of microbial cells that are communicating with each other through the chemical means of quorum sensing. And this allows them to basically offer protection to each other. That biofilm layer, that thin layer that they form, actually helps to create some resistance to things like antibiotics and soaps and detergents. So we find them on things like IV tubing, ventilators, anesthesia equipment, and it is a key opportunistic pathogen. It does produce a major virulence factor. And the major virulence factor that it produces is known as exotoxin A. And exotoxin A works to inhibit protein synthesis in the host cell. So it does have the ability to target and destroy the cell that it's infecting. It does have the ability, even though it is aerobic, it does have the ability in some circumstances to grow anaerobically. So we often find this with folks who have suffered second and third degree burns and have a breakdown of that integumentary system where some of those deeper tissues are now exposed. Patients who are cystic fibrosis patients and have weakened lung tracts are also able to be impacted significantly by Pseudomonas aeruginosa as well. There's also situations where this has been acquired through hot tubs and things like that as well. So complications can include a whole series of diseases, things like pneumonia, urinary tract infection, otitis, which is a inflammation of the eardrum, and corneal diseases. Significant symptoms can include endocarditis, which is an inflammation of the heart tissues, meningitis, which impacts the brain and spinal cord, and pneumonia. And as we mentioned, they do produce a kind of greenish blue pigment, which is known as pyocyanin. And that pyocyanin has the ability to give the microbes a certain degree of resistance. So we're going to pop in and start talking about some of the other gram-negative aerobic rods, specifically Burkholderia, Acinetobacter, and Stenotrophomonas. These are all very similar to the Pseudomonads in that they exist in a wide range of different habitats. They are obligate aerobes, and they're motile as well. So let's start off talking about the genus Burkholderia first. We've got two different species in this group, and they are both opportunistic agents in the fact that they are able to impact the respiratory tract, the urinary tract, and there are some that occasionally do cause skin infections. Burkholderia sapacea is really well known for its opportunistic infection of the respiratory tract. Burkholderia pseudomeli is generally acquired through some type of a break in the skin or inhaled from an environmental reservoir and can lead to things like bronchitis and pneumonia. We are also going to talk about septicemia later on, so it's really important to understand anytime you see this ending to a word that's emia, that involves that there's some type of a microbial organism circulating in the blood. So if it was bacteremia, it would be bacteria, or if it was viremia, it would be viruses. So this indicates that there is some type of microbial organism circulating in the blood and being able to travel to other organs and tissues in the body. So we're also going to talk about Stenotrophomonas, and this is a member of the fecal microbiota, and it's able to be isolated from domestic animals. So in clinical settings, again, this could be a contaminant of hospital equipment, water dispensers, and it's able to form the biofilms like we saw with Pseudomonas. And those biofilms help to protect it against the antimicrobial treatments. Again, it's opportunistic in that we usually see infections in patients who are suffering from cancer. And it is one of the most drug-resistant bacteria that we know of. Now we're going to get into talking about Brucella. And this causes a disease known as brucellosis. There are two different species, Brucella abortus and Brucella suis, which impact cattle and pigs respectively. Brucellosis has kind of two other names to it, either Bang's disease or undulant fever. And undulant fever means that you get this fluctuating pattern of fever over the course of several weeks to years. This is a zoonotic infection. Again, that it is commonly found in animals such as cattle, pigs, dogs, and goats. Now, there are some significant symptoms and other types of mechanisms that this pathogen uses to survive. So, brucellosis often causes swollen lymph nodes or swelling in the spleen. So, enlargement of the spleen and the lymph nodes. This is one organism where pasteurization becomes an important component to prevent infection. So, pasteurizing products from animals, especially milk, helps to reduce infections. There are a series of complications that can result from Brucella infection, including things like depression, meningitis, endocarditis, and a very deep-seated one that impacts the bones, known as osteomyelitis. So there is a vaccine for animals that is available for this. There are treatments, so it's a combination of tetracycline, which we saw falls into that protein synthesis inhibitor category, and rifampin or streptomycin. We're going to get into and talk about Francisella, and Francisella tularensis causes a disease known as tularemia, or very common name is rabbit fever. Again, this is a zoonotic disease. It's acquired from mammals, and it is endemic and consistently present in the northern hemisphere, especially in rabbits as the major animal category. It is transmitted through several means. Could be from contact with the infected animal, water, or even the vector itself. And the major vector that we see here are the ticks. The common symptoms seen with tularemia include headache, backache, fever, chills, and just general weakness. Will also see that with tularemia, we also get a swelling of the lymph nodes and enlargement of the spleen. Very similar to what we saw with Brucella, which also caused the swollen lymph nodes or spleen. These can survive phagocytosis just like Brucella. So if they are ingested by a phagocyte or a white blood cell, they are able to survive from being degraded. There is one major growth condition. So these are what we call fastidious, meaning that they have specific growth conditions that they require when we grow them on lab media. Francisella requires a source of the amino acid cysteine. So cysteine has to be added to the growth media. And cysteine is spelled, in case you're taking notes on this, cysteine is spelled Cysteine. And that is one of the common sulfur-bearing amino acids. And the pathogen has to have this in the media for us to be able to grow it. It causes a series of different major symptoms, ulcerative conditions at the site of the bite, swollen lymph nodes, as we mentioned, the swollen spleen. There's about a 10% death rate when we talk about the condition going systemic or impacting the lungs, the pulmonic form. There is no vaccine currently available, and for a while it was actually listed very high on the bioterrorism list. So we get this presence of this ulcer at the tick or insect bite. Bordetella pertussis is the causative agent of whooping cough. And it's important to note that we have seen a gradual decline of pertussis infections since 1976. And a lot of that is just because of a very rigorous and proactive vaccination campaign for young children. This is a Coccobacillus. When we talk about a Coccobacillus, this is a morphology where you get these kind of short, plump rods. So that it's kind of a fusion between a spherical coccus and a elongated rod-shaped cell. So you get these kind of short, plump rods. It does possess a capsule. And the capsule, again, is this outer layer that works to protect from phagocytosis. This is a childhood affliction, respiratory syndrome that is very severe in very young children. And it's, again, they just don't have the immune response mounted to be able to fight this infection off. Transmission can occur either through direct contact with a healthy carrier adult, or it can be from inhalation of aerosols. Coughs and sneeze can also transmit the pathogen. So we have seen pertussis incidence has kind of started to rise in the years, even though as a general whole, we've seen a decrease in infections since the late 70s. A lot of this, again, is from folks who are just not looking at the vaccinations as an option. And it's impacting greatly children who are under 1 year of age. Otherwise, very low incidence for other age demographics. So, virulence factors, one of the big things is they produce toxins, and these toxins are able to bind to and destroy your ciliated cells in your respiratory tract. So, on a lot of your mucus tracts in your body, the cells possess cilia. And remember that cilia can actually beat back and forth. And as they undulate back and forth, they help to move mucus and other materials through your tract. Well, when the toxin goes in and it destroys the ciliated cells, it actually causes mucus to begin to accumulate and eventually block your airways, leading to suffocation. There is a vaccine, and the vaccine is one that includes vaccinations to other diseases as well. It's known as the DTaP vaccine, and that goes after diphtheria. Tetanus is the T, and acellular pertussis. So, acellular basically means that they are just taking the toxin, pieces of the toxin, and other antigens to mount an immune response. There are two stages that I do want to note as well when we talk about pertussis. We have the first stage, which is known as the catarrhal stage. The catarrhal stage lasts between 1 to 2 weeks, and then that is soon followed by the typical stage where we get the severe barking cough. That's known as the paroxysmal stage. We have the catarrhal stage, which is about 1 to 2 weeks in length, followed by the stage with the severe cough, known as the paroxysmal stage. The genus Alkalogenes is one that has the potential to be part of our normal flora. It is one of the most common clinical species found in places like the feces, sputum, and urine, and it is occasionally associated with opportunistic infections, things like pneumonia, septicemia, and meningitis. Legionella pneumophila, which we mentioned earlier, is the causative agent of a disease known as legionellosis or Legionnaire's disease. It was first documented in the year 1976 at an outbreak of the American Legion Union, where about 200 people caught the infection, with 29 dying from it. And it was actually, they had tested the food, they tested the water at the convention, and ended up finding out that it was being acquired from the air conditioning system in the building. So this particular pathogen is often associated with waterborne aerosol particles from things like air conditioning units, and even the produce misters at the grocery store have been known to spread cases. Again, it's widely distributed in water towers, air conditioners, misters in the produce section at the grocery store, and it is a combination of Legionnaire's disease, which is the most severe, but also a less severe infection known as Pontiac fever. It is extremely prevalent in males, more common in males than females, and targeting those that are 50 years or older in age. Cases of this infection have even been able to be traced back to the Mount St. Helens volcanic eruption. And the common symptoms with this include things like cough, diarrhea, abdominal pain, fever, and the fatality rate from the pneumonia is between 3 and 30% in Legionnaire's disease. Now we're going to move into and start talking about the Enterobacteriaceae, which are a large family of organisms that include things like E. coli and Salmonella. These are gram negative, and they are also non-spore forming rods. And we're going to see that a lot of these are targeting places like the soil, water, and also decaying matter. We also find them as members of the large intestine in animals, and the most frequent cause of diarrhea through toxin formation. The Enterobacteriaceae are also all glucose fermenters. So all of the members of this family are able to ferment glucose. So I also want to mention this is an important slide to be aware of. A lot of the organisms that are on this slide fall into this family, but characteristic, they are opportunists, meaning that they will only infect when the immune system is weakened. So things like E. coli, Klebsiella pneumoniae, Enterobacter aerugineus, Serratia marcescens, Proteus, and Citrobacter all fall into this family, but again, are only going to create a problem when the patient is an opportunist. So we've already mentioned things like glucose fermentation. All the members in this family do this. They're able to reduce nitrates. They are oxidase negative. Most have the presence of flagella for movement. And again, they're all rods, gram-negative rods. We can further divide this family into two smaller groups. We have the coliforms, which are lactose fermenters, so that's important to know. Coliform implies that they ferment lactose. And obviously then the non-coliforms would be non-lactose fermenters. We talked about in one of the first chapters in the course, there's actually a book known as Berge's Manual of Systematic Bacteriology. It's a multi-part series that basically walks through these tools called dichotomous keys. And these dichotomous keys help you after you perform a gram stain, which is usually the initial test in any identification of a microbe, an unknown microbe. We can then go into Berge's manual and use a series of these dichotomous keys where you basically go yes or no, and then depending on which option you choose, you follow these brackets down until you get to an actual microbial identification. But all of these different items that you see in pink are different tests that you would perform on laboratory media to basically give you an idea of which unknown microbe you were working with. So I wanted to point that out to you that we actually use a series of already developed results to figure out what pathogen we are looking at based on a lot of the biochemical identification, those physical methods that we use to identify microbes. We will get into that a lot more in chapter 17. We'll start talking about things like the genotypic, phenotypic methods and the biochemical tests that we use to identify unknown microbes. You do not need to know for testing. Table 23, I am not going to ask you any of this information on an exam. More just an FYI to show you how a lot of these different pathogens differ on different types of biochemical media. This slide is extremely important, however, to know. There are antigens that are displayed on the surface of these Enterobacteriaceae that contribute to virulence and the ability to trigger an immune response. So there are three antigens that we often describe with a letter. The first one is the H antigen, and this is the one that indicates the type of flagella for movement. The K antigen is the capsule, and also can be the Fimbriae antigen as well. And then the O antigen is the cell wall antigen. So make sure that you're familiar with the H, K, and the O. H is for the flagella, K is for the capsule, O is the cell wall antigen. And not all species are going to carry the H and the K antigens. But all of these, since they all have a cell wall, since they're gram negative, they have that thin peptidoglycan cell wall, they are all going to possess this O antigen. The pathogenesis of these enterics as well is also going to rely on the production of toxins, endotoxins or exotoxins. So whether it's from the LPS, from the outer membrane as an endotoxin, or a toxin that's secreted, an exotoxin. We're going to talk about the AB toxins in a few moments, but those are examples of exotoxins. So again, just a picture, capsule antigen is our K. It's that surrounding layer that protects the cell from phagocytosis, allows for attachment in some cases. We have the O antigen or the cell wall antigen. And then we also have our H antigen or flagella antigen. And again, they have the opportunity to possess an H and a K, but all enterics are going to have this O antigen for their cell wall. So let's talk about the most common of all of the enteric bacilli, and that being E. coli. E. coli is significant because about 70% of all travelers' diarrhea originates from the enterotoxins that are produced by E. coli. We have about 150 different strains that we know of, some being opportunistic, some being normal flora, meaning that they live in our gut as protective bacteria in our intestines. The pathogenic strains can be grouped in a couple different ways. So they can be what we call enterohemorrhagic, EHEC strains. And these cause conditions such as HUS. So the enterohemorrhagic strain can cause a condition known as hemorrhagic syndrome, which can cause damage to the kidneys. We have the enterotoxigenic E. coli, which again can also produce diarrhea due to the production of a toxin that is heat stable and can cause the host cells to lose fluids. We have the enteroinvasive, which causes inflammation in the large intestine. Enteropathogenic is linked for infants in a wasting disease, meaning where they basically just start to lose weight and body mass. The enteroaggressive form is the common cause of pediatric diarrhea. And then we also have what's known as the diffusely adherent E. coli or DAKE, and these are a very minor cause of pediatric diarrhea. So what I want you to pull away from this is when we're talking about the E. coli and we're talking about all of these different forms, I want you to notice that they all have some type of diarrhea associated with them. Okay, so things like the enteropathogenic, the enteroagglutinative, the diffusely adherent E. coli, and our enterotoxigenic E. coli. So we mentioned that about 70% can cause the traveler's diarrhea, the bloody stool. 50 to 80% of UTIs are also caused by E. coli as well. And again, a lot of this has to deal with the physical anatomy between males and females. Again, the opening of the digestive tract and the opening of the urinary tract are relatively close together, allowing for fecal bacteria to often be pushed into other body cavities, allowing for these infections to grow up. Oh, and I do want to mention bismuth salicylate or Pepto-Bismol. The reason why Pepto-Bismol, the pink stuff, works so good is it actually goes in and can counteract enterotoxins, those digestive toxins, which will reduce the symptoms and stop the inflammation and other types of cell damage that are occurring. I do want to talk about one very important member of the E. coli family, and that is the O157H7. So again, remember the O and the H refer to antigens. So it has flagella antigen type 7 and cell wall antigen type 157. This is a enterohemorrhagic strain. So as we mentioned, it is responsible for causing this HUS, this hemorrhagic syndrome, which can lead to kidney damage. Again, HUS stands for hemorrhagic uremic syndrome. And it was first noted in fast food hamburgers. There was an outbreak known as the Jack in the Box restaurant in 1982, where several people consumed hamburger meat that day and acquired this infection. The course of my graduate work, when I was at the University of Rhode Island, I was working with studying the impact of nutrient utilization by both commensal E. coli and pathogenic E. coli. And my work actually helped to support the beginnings of the probiotic era, where we studied the ability to have beneficial bacteria colonize the intestinal tract and actually reduce the ability for pathogens to grow at that body region. So, a lot of my work and publications have actually come from working with E. coli. The toxin is able to enter the host cell. It binds to the ribosomes, stopping protein synthesis, and obviously that leads to death as well as destruction of your intestinal cells. We also talk a lot about something called the AE lesions. The AE lesions can cause intestinal damage because it actually creates these pedestals that form from actin rearrangement in the intestinal cells. Those are known as A/E lesions. The inflammatory disease that happens from E. coli infections is very similar to those seen with the bacteria Shigella. So currently, we test our commercial meat on a random basis, the USDA, to examine for the presence of microbial contamination as well as safe handling and packaging processes prior to purchase by consumers. We do have some other coliforms. Again, remember that these are less common than the E. coli, but also are opportunistic in nature. There's Klebsiella pneumoniae, which is commonly a normal inhabitant of the respiratory tract. It has one of the most pronounced capsules that we know of on a bacterial organism, and it does cause a range of nosocomial infections, pneumonia, meningitis, UTIs. Enterobacter, again, is also found with UTIs and surgical wounds. Citrobacter, again, UTIs. Serratia marcescens actually produces a heat labile pigment, a red pigment, that expresses only at certain temperatures. When we grow the microbe at certain temperatures, it has the ability to give off a red hue to its colonies. We see serratia a lot with burn and wound infections, resulting in things like septicemia and meningitis. So here's an example of that serratia, which produces these colonies with a red hue to them. You'll also notice a lot when an organism produces a capsule, it gets this kind of sticky looking, shiny texture to it that's resulting from that capsule production. So we've just talked about the coliform bacteria, members of that Enterobacteriaceae family. And those, again, the reason that they're called coliforms is for their ability to ferment lactose. We're now going to talk about the non-coliform negative gram-negative bacteria. So these do not ferment lactose. We've got a few that fit into this category. We have the opportunists, things like Proteus, Morganella, and Providencia. And then we'll focus a lot of our time on the two enteric pathogens, which are true pathogens, Salmonella and Shigella, which are similar but have some key differences between them. So ordinarily, these are extremely harmless. They are saprobes, which lead to degradation of organic matter and can also be part of our intestinal tract for humans and animals. Proteus have this kind of swarming appearance when they're growing on an agar surface, and we often find them involved in UTI infections is the major area that we see Proteus. Proteus vulgaris is the organism that we're talking about. Again, UTIs, their common causative disease. Morganella and Providencia are also found in similar infections such as UTI, pneumonia, and wound infections as well. The true enteric pathogens, on the other hand, include Salmonella and Shigella. And these are extremely common. They have extremely well-developed virulence factors that allow for colonization and invasion to the body. And they are not part of the normal human flora, so these are true pathogens. They do have a significant gastrointestinal involvement and cause a form of diarrhea. However, they do have, in some cases, the ability to become septicemic and travel to other systems of the body. So the genus Salmonella contains really two big species, Salmonella bongori, which is found almost exclusively in cold-blooded animals, and Salmonella enterica, which is divided into 6 subspecies. And there are about 2,500 serotypes that can be identified from these different pathogen groups. Again, these are flagellated. They are motile, so that is extremely important to know. So the Salmonella do have the ability for movement, and they are extremely resistant to chemicals such as bile and dyes. And that's important since they are in the digestive tract and targeting those organs. They want to be resistant to things like bile. Salmonella, as we mentioned, is a true enteric pathogen. It's zoonotic, and its drug resistance continues to increase. We see more and more cases of drug-resistant salmonella in the population. We see that its common outbreaks come from things like poultry and exposure to eggs. So eggs and poultry are two of the major sources of salmonella infections. They do have the O and the K antigens. However, the O antigen during testing can sometimes be masked or hidden by the K antigen. So it makes identification difficult in some cases. I also do want to mention to you that when we're talking about these villi, these folds in the intestine, Salmonella can actually break free from these intestinal folds and actually get into the bloodstream and be carried to other places in the body. So we have seen a growing and then steady number of cases of typhoid over the last 40 years or so, which is significant in all sorts of different food products, from cookie dough to eggs to meat. So it's really caused the USDA to look at safe food handling and packaging procedures. We also want to talk about another member of the Salmonella family known as Salmonella typhi. And Salmonella typhi causes a disease known as typhoid fever. It is acquired through fecal contaminated food or water. So it's from ingesting those. And for most folks, they are asymptomatic carriers. And it's really important to know that these asymptomatic carriers actually harbor the Salmonella organism in their gallbladders. The Salmonella typhi organism does have the ability to infect the small intestine. And when it does infect the small intestine, it's going to cause things like fever, diarrhea, and abdominal pain. But it can become septicemic. So it can spread to the lymph nodes, the spleen, It can even cause abscesses to form on the liver. Again, not a coliform. This organism does not ferment lactose, and it does have the ability to be able to enter into phagocytes and survive within those phagocytes and not be destroyed by them. So now that we've talked about Salmonella, we're going to look at a very similar related species known as Shigella and talk a little bit about the differences between Salmonella and Shigella. Very similar to Salmonella, one of the things that they do have in common is Shigella is also a true enteric pathogen. So these are not found in the normal flora. The first key difference is they are non-modal. They do not exhibit movement. So that H antigen is absent in Salmonella. They do not have the flagella antigen. If they're going to move from cell to cell, they actually utilize the actin tails to do that movement. There are 4 big members in this particular family. There's Shigella dysenteriae, Shigella soniae, Shigella flexneri, and Shigella boydii. Again, so we saw that Shigella, just like Salmonella, is a true enteric pathogen. But unlike Salmonella, which is able to break out of these intestinal villi and travel to other sites in the body, Shigella invades the large intestine villi, but does not perforate the intestine or travel to the blood. So it stays localized to a particular place known as the Peyer's patches and causes inflammation from the production of not only endotoxins, but also exotoxins as well. The exotoxin is known as the AB toxin. So the AB toxin, the A portion is the attachment. That's the toxin piece that's going to attach it to its location. And the B portion, or excuse me, the B portion is the attachment, the binding portion. The A portion is the active portion. So that's the A portion is what's going to actually cause the damage to that particular region. And you actually get the production of these bloody mucus stools. They call it bloody mucoid stools. As we mentioned, human carriers are going to transmit through fecal matter, contaminated fecal matter. And it's often called, when we talk about Shigella, it's often called bacillary dysentery. And we've seen large outbreaks in places like daycares with this particular organism. So again, no perforation of the intestinal tract. They're going to stay localized in those intestinal villi. We're also going to talk about the Yercinia. Yercinia is another genus of enteric pathogens. And specifically, we're going to get into a few different types. We're going to talk about Yercinia enterocollitica, Yercinia pseudotuberculosis, and then the most common that we are going to talk about is Yercinia pestis, which causes the plague. So when we talk about Yercinia enterocollitica, this reservoir is usually in places like domestic and wild animals, different types of fruits and vegetables, and fish. The bacteria get into the intestinal mucosa and can also get into places like the lymph nodes and survive within the phagocytes. The symptoms actually mimic the pain that is seen with appendicitis attacks. Yersinia pseudotuberculosis, similar type of pathogenicity, but it's really targeting the lymph nodes in this case. Yersinia pestis is the major one that we're going to talk about, and this is also known as the plague, or most commonly called in the 1300s, when this was pretty relevant, was the Black Death. In the 1300s, about 25% of the population in Europe actually died from the plague. So this was a extremely relevant disease for those populations during this time. It is non-enteric, and these are also gram-negative rods, and they have several virulence factors to them. They have capsular and envelope capabilities, but they also produce some enzymes as well. Coagulase, which is able to stop clotting from occurring. They do have an endotoxin from the outer membrane, that LPS component, and they also have something known as the murine toxin as well. So when we talk about the plague, humans can actually acquire it through contact with animals, domestic or semi-domestic, and there's about 200 different species of mammals, particularly the rodents, that are able to harbor this infectious disease. The flea is the major vector that is responsible for the spread from the rodents to humans. And the flea, when it takes a blood meal and actually acquires the pathogen, it begins to replicate in the gut of the flea. However, that coagulase enzyme is then able to cause blood clotting that will block the esophagus. And basically, what happens now is since the esophagus is blocked and the flea can no longer eat to fill its stomach, it becomes ravenous and starts to bite everything in sight, trying to take a blood meal, and in that process spreads the pathogen. So we see a lot of times, again, our rodents, things like squirrels, chipmunks, and mice are the major rodents responsible for harboring the disease. Flea is the vector that's carrying it from one host to the next. We are going to talk about the three forms of plague. We have the bubonic plague, the pneumonic plague, and we also have something known as the septicemic plague as well. So let's get into the three different forms. The infectious dose is relatively small, only about 3 to 50 cells to actually cause infection. And the symptoms typically arise between 1 to 6 days after exposure. The bubonic plague is commonly known because it forms these massive swelling of the lymph nodes, these almost like tumors that form, known as buboes. And these buboes form in the armpit region or in the groin region. The pneumonic plague is the form that the sputum is actually contagious during this particular phase, and the infection is localized to the lungs, extremely contagious and extremely fatal if left untreated. The septicemic plague, on the other hand, is one where the organism starts to spread throughout the body, and this is often the form that we refer to as the Black Death. Its virulence factors actually cause your intravascular system to coagulate, and you get this hemorrhaging under the skin or this kind of black hue to the skin known as purpura. So it's often sometimes called the Black Plague. Um, Obviously, treatment with this is reducing the rodent population. If we reduce the rodents, that in turn is going to limit the spread. The presence of that capsule and that coagulase enzyme are really important. Again, the capsule helps to reduce the phagocytosis, and that coagulase enzyme, as we talked about, helps in the process of forming those clots. We see the swollen lymph nodes, we see the enlarged spleen, just like we've seen in some of our other infections that we've talked about in this chapter. Francisella, tularemia, and Brucella, as well as Yercinia, do all result in the swollen lymph nodes or enlarged spleen. Alexandra Yersin was the first person to really discuss the vaccine to this infection in the year 1866. So about 500 years after the original outbreak of this disease, Alexander Yersin came up with its first vaccination. So diagnosis again is going to depend on the signs, the symptoms. Treatments are antimicrobial, streptomycin, tetracycline, or chloramphenicol. And as we mentioned, Yersin came up with the first vaccine in 1866. And reduction of the rodent population is really one of the main mechanisms to stop the spread of this pathogen. In terms of lab results, staining is one of the key tests, and again, they do what's known as a bipolar stain to determine the presence of Yersinia. We're going to talk about the oxidase positive organisms now. The first one being Pasteurella multacida, is a zoonotic microbe from animals. And there are 6 species that are most common in this group. Obviously, Pasteurella multacida, which is the largest of the opportunistic infections. But then we also have several that fit into this genus Haemophilus. Haemophilus influenzae, which is probably the most common one that we see there. We will talk about some of these other ones today as well. So Pasteurella multacida is significant because it is acquired through dog and cat bites or scratches. And these abscesses actually form at the site where the dog or cat scratch or bite occurred. And these organisms can then spread to other regions of the body, like the joints, the bones, and the lymph nodes. Immunocompromised folks are the most common for severe infections with this particular infectious disease. So we also talk about the Haemophilus, and we're going to really focus on 2 of the members of this group today. We're going to talk about Haemophilus influenzae and Haemophilus ducreyi. Haemophilus influenzae is the causative agent of a whole slew of different infections. Bronchitis, epiglottitis, which is an inflammation of the epiglottis, meningitis, and also otitis media, that middle ear infection. These are tiny gram-negative rods. They're pleomorphic, which means they can vary their shape. Fastidious again, meaning that they require really specific growth conditions. And you'll notice down here that they actually require in their media things like hemin, NAD, and NADP. These are electron transfer components. So if we're talking about the electron transport chain, these are electron shuttle vectors. And we often grow them on a media known as chocolate agar. And chocolate agar is where we would take and lyse the red blood cells to release the heme and into the agar. So it gets this almost milk chocolate appearance to the color. Many of the species are normal colonists of our upper respiratory tract or vagina. Things like Haemophilus parainfluenzae or Haemophilus ducreyi. And we will talk about, obviously, we get into and we talk about things like Haemophilus ducreyi. This is responsible for a disease known as cancroid. So the Haemophilus are often termed the blood-loving bacilli. These are the ones that require those components from the red blood cells. Haemophilus influenzae, again, we mentioned, causes things like meningitis, epiglottitis, otitis media, sinusitis, and then things like bronchitis. There is a vaccination. Haemophilus influenza type B has a vaccination known as the HIB or HIB vaccine, which is commonly given in combination with the Tdap vaccine, the tetanus, diphtheria, and pertussis. And since the vaccine was introduced in the early 1990s, we've seen the decrease of cases almost to the point where it's non-existent in the population. The Haemophilus influenzae aegyptius subtype is commonly responsible for the disease pink eye or conjunctivitis, which is an inflammation of the conjunctiva. We have talked about Haemophilus ducreyi, which is a sexually transmitted infection known as cancroid. And this is significant because it produces this bubo-like swelling at the lymph nodes, similar to what we saw with plague. You also find lesions produced at the portal of entry. However, the infection is really regionalized to the tropics and subtropics. We also have Haemophilus parainfluenzae and Haemophilus afrophilus, which are members of the normal flora and are seen in cases of endocarditis with the heart. So I do want to end today and just talk briefly about septic shock. So one of the big things that we often hear is the presence of septic shock. And septic shock is results from the presence of that LPS. So in the outer membrane, we have that lipopolysaccharide component. The lipopolysaccharide component is able to break free and travel in the blood and cause a series of symptoms such as tachycardia, respiratory failure, weakened pulses, but most significantly, in septic shock, you actually see a reduced blood flow to your vital organs. And the reason for that is there's actually blockages in the very narrow capillaries that prevent blood flow and create this condition of hypoxia, where there's a lack of oxygen to your vital organs. So that's really how you eventually perish from septic shock. I also want to just clarify that when we talk about sepsis, sepsis is different from bacteremia. So anytime we talk about any of the emias, bacteremia specifically, bacteremia involves the fact that there is a actual living bacteria circulating in the blood. Sepsis, on the other hand, implies that it's only the toxins, specifically the endotoxins from the gram-positive or gram-negative bacteria, are circulating in the blood. And those endotoxins can actually travel to the lungs and cause lung damage in patients, which ultimately leads to the cause of death. So, with that said, that is the wrap up for chapter 20. If you have any questions as you're reviewing, please feel free to bring them to office hours. I recommend highly to you, please make sure that you are creating some type of a table or chart to organize your information. I would definitely focus on the following. I would make sure that you create a chart for each chapter, saying whether it's a gram negative, gram positive, bacillus, coccus, whatever the shape for that chapter is. And then in your chart, I'd have a place for the pathogen name, the disease it causes, the symptoms. So, what are the key symptoms? And anything significant about how it is grown. So it may be the reservoir and the environment it comes from. It may be the type of lab media if it requires a particular set of nutrients to grow. So any of those key characteristic things that we've walked about. So disease, causative agent, symptoms, and any specific information about its virulence, its ability to grow on lab media. Those are important things that will come back in testing purposes. So thanks, everybody. I look forward to seeing you in the discussion board or in office hours, and hope all is well. Take care.